Welcome to another physio video. Let's talk about the synthesis of thyroid hormones. Remember from the first video on the thyroid that it's the follicular cells that make thyroid hormone. To understand how these cells do this, let's zoom in on a follicular cell and a capillary. Here is a zoomed-in view of the follicular cell. Let's label some important parts of this image. The lumen is the inside space of the thyroid follicle. The membrane closest to the blood is the basolateral membrane. The membrane closest to the lumen is the apical membrane. And don't forget the capillary. The two hormones made by the thyroid gland are T3 and T4. Several steps are involved in making T3 and T4, but let's start with a very important ingredient called the Glycoprotein Thyroglobulin, abbreviated TG. Thyroglobulin is synthesized in the nucleus and rough endoplasmic reticulum and then packaged into a vesicle inside the Golgi apparatus to then be secreted into the follicular lumen by exocytosis. On the basolateral membrane of the follicular cell, there are sodium-potassium pumps. Remember that this pump uses ATP to pump 3 sodium ions out of the cell for every 2 potassium ions that are pumped into the cell. Sodium-potassium pumps create a gradient for sodium ions that provides the energy for a carrier called the sodium iodide symporter, or NIS. Remember that with secondary active transport, there is one ion that goes from high to low to provide the energy for another ion or molecule to go from low to high. Sodium enters the NIS and goes from high to low, and provides the energy for iodide to go from low to high and be pumped across the basolateral membrane. Both ions cross going the same direction, so it's called symport. There is a protein carrier called pendrin located on the apical membrane that transports iodide from inside of the cell into the lumen of the thyroid follicle. Also on the apical membrane, there's an enzyme called Thyroid Peroxidase, or TPO. Inside the lumen, TPO oxidizes iodide or causes it to lose an electron to become iodine. Iodide being oxidized to iodine facilitates its combining with tyrosine molecule located on the thyroglobulin. One iodine combining with one tyrosine results in the formation of a monoiodotyrosine, or MIT. When two iodines combine with one tyrosine, a diiodotyrosine, or DIT, forms. TPO will then combine MITs and DITs to create the thyroid hormones T3 and T4. T3 forms when a MIT combines with a DIT. T4 is created when 2 DITs are added together. T4 is called tetraiodothyronine because it has 4 iodines. T4 is the inactive version of thyroid hormone. About 90% of the thyroid hormone inside the thyroid follicles is in the form of T4. The remaining 10% of thyroid hormone is in the form of T3. T3 is the active form of the hormone and has 3 iodines, so it's called triiodothyronine. Our newly created thyroid hormones must now gain access to the blood so they can be distributed throughout the body. To do this, iodinated thyroglobulin is taken into the follicular cell by way of receptor-mediated endocytosis. The receptor for this endocytosis is called Magellan. Once the thyroglobulin complex is in the follicular cell, a lysosome will fuse with the vesicle and begin to break down the thyroglobulin complex. The lysosome breaks down the thyroglobulin complex into mits and dits, amino acids, and T3 and T4 hormones. In the cytoplasm of the follicular cell, there is an enzyme called intracellular deiodinase. This enzyme removes iodines from mits and dits. The iodines can now be recycled and used in the process to make more thyroid hormones. The amino acids can also be recycled to make more thyroglobulin. Now back to the thyroid hormones getting to the blood. On the basolateral membrane, there is T3, T4 carrier protein that transports T3 and T4 across the basolateral membrane so that they can get into the blood. Inside the capillary, T3 and T4 bind to thyroid binding globulin, or TBG, for transport in the circulatory system throughout the body. In peripheral tissues, T4 is converted to active T3 by an enzyme called peripheral deiodinase. T3 then binds to its receptors and exerts its action to increase growth and metabolism. An important hormone for the production of T3 and T4 is thyroid stimulating hormone, or TSH. That is produced and released from the anterior pituitary gland. The receptor for TSH is located on the basolateral membrane of the follicular cells and binds TSH. This receptor-ligand interaction results in an increased production of cyclic AMP from ATP. Increased activation of the TSH receptor will ultimately lead to increased levels and activity of sodium-potassium pumps, NIS, TPO, pendrin, thyroglobulin, Magellan, T3, T4 transporters, etc., that all act together to increase the production and release of T3 and T4 into the blood. Regulation of thyroid hormones ultimately starts in the hypothalamus with the release of thyroid-releasing hormone, or TRH. TRH travels a short distance in the blood from the hypothalamus to the anterior pituitary gland via the hypothalamo-hypophyseal portal system. It then stimulates anterior pituitary cells to release thyroid-stimulating hormone, or TSH. TSH then enters systemic circulation and travels to the thyroid gland to stimulate TSH receptors that we just discussed to increase production of T3 and T4. All of these hormones are regulated through a negative feedback system that allows them to stay in normal range and not get too high or too low. This concludes the video explaining thyroid hormone synthesis. Thanks for watching.